My name is Hogue Levins, and I'd like to welcome you to this segment of Asbestos Safety Workshop. Asbestos Safety Workshop is a series of features in which we explore the latest tools and technologies of the asbestos safety industry. And today, we're going to be talking about testing. That is, we're going to see how you study a building, whether it's a single home or a 100-story skyscraper, to determine if asbestos hazards are present there. Knowing the exact location and quantity of asbestos present in a building is the key for assessing health hazards. This information is also a crucial part of the evidence needed for filing recovery or other asbestos in buildings related lawsuits. The process of accurately testing buildings for asbestos hazard is one which calls for common sense as well as some fairly involved technical procedures. During this program, we're going to be taking you step by step through those procedures. We're going to be interviewing some national authorities in the field of asbestos testing, and we're going to be taking you inside laboratories so that you can actually see as asbestos samples are prepared and analyzed by various kinds of microscopes. Before we get down to the nuts and bolts of all that, let's look outside our studio and get a better perspective on the general asbestos in buildings problem. This skyline just west of our studio is Philadelphia, America's fourth largest city. It's a very pleasant place which has the same problems as other urban areas across the country. And as you can see, the Philadelphia skyline, much the same as the skylines of New York or Chicago or San Francisco, contains buildings of every imaginable size, shape, and age. And something like 40% of these buildings have asbestos of one sort or another inside them. During the last 2 decades, health authorities throughout the Western world have been warning about the cancer and other disease dangers posed by asbestos. In 1978, the federal government's top health authority issued an unprecedented national warning about asbestos dangers. And although the public concern on this issue came to focus largely on school buildings, researchers were finding that airborne asbestos posed a danger in buildings of all types. In the summer of 1984, the U.S. Environmental Protection Agency completed a nationwide study and reported that as many as 700,000 buildings in cities across the country have potentially dangerous asbestos in them. Many authorities feel that the EPA figures are very conservative. And these 700,000 buildings cited by the EPA don't include the hundreds of thousands of homes which have asbestos in them and can also expose their occupants to serious health risks. How do you know if you have an asbestos problem in your building? What's an asbestos problem look like? How do you recognize it? How do you make judgments about its severity? Well, that's a big part of the problem because with asbestos, the problem is invisible. The kinds of fibers which are most dangerous to the human body and which are most prone to be airborne to the human eye. And as a matter of fact, if the room I'm standing in were contaminated with airborne asbestos, the air would look exactly as it looks now, perfectly clear. Let's go back into our studio and look at some asbestos and try to understand the dynamics of this problem a little better. Asbestos is a mineral, a kind of rock, but a very weird kind of rock, and to understand why, let's look at an ordinary rock. This is your standard rock that you might find laying around outside, and one of its characteristics is that if we take a hammer and we bash this rock up into the smallest particles possible, what we'll end up with is this, your common everyday sand. Well, one of the characteristics of sand is that no matter how much you pound it up and break it down smaller and smaller, each one of the little particles in this dish will always hold a shape similar to this, roundish. And the reason is that the crystalline structure of ordinary rocks always breaks down into relatively round, lumpish crystalline forms similar to this. Asbestos rock, on the other hand, is very different. It's not formed like ordinary rock. Instead, it has veins of tightly packed rock fibers. And when you pulverize asbestos rock, you get this, a fibrous mass. And no matter how much you pulverize this material, it will continue to break down into smaller and ever thinner forms, like this. The elemental differences in shape between the normal lumpish particles of ordinary rock and the long, thin, fibrous particles of asbestos rock is what makes asbestos bizarre among minerals. No other substance will hold this very long, very thin, and very strong shape. Now, many people, when they hear the word fiber, tend to equate that word with something soft like yarn or sewing twine. Well, in fact, the fibers of asbestos aren't soft at all. They have the tensile strength of steel, and they are much more like needles than anything soft like yarn. This tightly sealed bottle holds asbestos-containing material which was recently removed from the ceiling of a New Jersey office building. If I would open the cap on the bottle, submicroscopic fibers would immediately begin floating into the air where I could breathe them. And as we'll see in a moment, these fibers can be very damaging to the interior of the human body. We now know there are at least 30 million tons of this kind of material installed in buildings across America. Up until the 1970s, asbestos was as common a construction material as was plywood or concrete, and it was installed in at least 50% of all the buildings put up across this entire country. Asbestos was added to thousands of different construction products. It was sprayed onto steel girders, it was mixed into plaster, pressed into wallboard, wrapped around pipes and slathered onto boilers, heating ducts, and utility systems, sprayed onto ceilings, walls, and other surfaces, and used in many other ways. To better understand why asbestos was used so extensively in construction, let's imagine that I have a handful of sand and I heave it at the ceiling. Well, what's going to happen is the sand's going to fall down on my head. However, now let's imagine that I have another handful of sand and I add a significant quantity of asbestos fiber to it. And to that I add another small amount of a glue-like binder and I mix them together and I heave that at the ceiling. Well, it's going to stick. And the reason is that asbestos becomes the matrix which holds together concrete or plaster or insulation-like materials. What happens is very much like when you add straw to mud to make bricks, except it happens on a very, very much smaller scale. Structurally, asbestos is very much like manila rope. Similar to manila rope, it has very large strands which are intertwined and can then be broken down into smaller strands, which can then be broken down smaller. And with asbestos, you eventually get to a point when the smallest strands you've broken down are invisible to your eye. This is a yardstick, and the metric equivalent is a meter, which is about the same length. And this is pretty much the way that we measure our world, in inches, yards, meters, millimeters. In fact, when most of us think about things that are small, we're thinking in terms of a fraction of an inch or a fraction of a millimeter. But to fully appreciate the dangers of asbestos in buildings, you have to learn to think in an entirely new dimension of smallness. And to give you an idea, the unit of measurement that scientists use to measure asbestos fibers that are dangerous is the micron. And if I took this stick and I was able to slice it down into 1 million parts, one of those parts would be a micron or a micrometer. A micron or a micrometer is 1/1,000,000th of a meter. When we take one of the visible strands in our bottle and magnify it under a scanning electron microscope, you can begin to appreciate the extraordinary dimensions of smallness involved with asbestos. This is a magnification of 240 times, and you can see what look like bundles of fibers and individual fibers of asbestos. When you take one of the smaller fibers you can see in this magnification and blow that one fiber up 6,600 times, this is what you see. What appeared to be a small fiber is not a fiber at all, but another bundle of smaller fibers. And if we again use the scanning electron microscope to magnify one of the individual fibers in this bundle 20,000 times, we see that even that isn't a fiber at all, but an unbelievably tiny bundle of yet smaller fibers. Each of these fibers are about 0.1 micron in diameter, but even this isn't the end. If we use a transmission electron microscope to blow up one of these 0.1 micron diameter fibers, we see that it's composed of what are known as fibrils, the smallest units of asbestos. And you can see fibrils have hollow centers. They're actually shaped like rolled up rugs, which are about 0.02 microns in diameter. This is a magnification of a typical mix of asbestos, plaster-like sealing material, and binders. You can easily see the large bundles of asbestos sticking every which way as they form the matrix which holds the stuff together. But because organic binders naturally deteriorate, billions of these tiny, invisible fibers can be released into the air spaces of a building and breathed in by building users. Asbestos in this state is called friable. That is, it's releasing invisible fibers into the air. In the publications it's issued on this subject over the last 10 years, the U.S. Environmental Protection Agency has placed a heavy emphasis on the concept of friable asbestos materials. But authorities in the field caution that the term is often confusing and misused. Anthony Natal, president of the Dual Corporation of Cherry Hill, New Jersey, and co-author of the industry reference book Asbestos Removal and Control, explains.
The friable conditions that are mentioned by EPA and OSHA sometimes can lead you astray. A very hard material can release very thin, long fibers, and vice versa. A very soft material releases very few. It's very hard to tell without the testing. You need the testing. It's your only eyes to see what you're doing, to see if you've actually accomplished what you were contracted to do. And without that testing, you would possibly be leaving a lot of people in jeopardy.
One problem, for instance, is that EPA publications using pictures like this have fostered the illusion that friable asbestos materials must be visibly falling apart before they present a danger to building occupants. Well, this is not the case. Let's take a look at an actual sealing situation. This is the ceiling of an office building. The ceiling is covered with a typical sprayed-on asbestos material, and notice that this ceiling is not visibly damaged. In fact, it looks pretty nice. However, when a scanning electron microscope is used to inspect the surface of this sort of ceiling, this is what you see. At 400 magnification, asbestos sealing materials begin to show their fibrous, friable nature. At 1000 magnification, you can begin to see the frayed ends of individual bundles of asbestos in the process of breaking down. At 6000 magnification, you can clearly see the tangle of asbestos fibers throughout the sealing material. The diameter size of most of these fibers is about 0.1 micron, and the mere passage of air currents across such a sealing surface can cause billions of these fibers to slough off and invisibly contaminate a building. For people who think in terms of meters and inches, something which is 0.1 micron in diameter is just about impossible to envision. 0.1 micron is 1/10 of 1/1,000,000th of a meter. But this incredibly tiny size is ultimately what makes asbestos so dangerous. No other substance, not even fiberglass or rock wool, will break down into fibers of such incredible thinness and strength. This scanning electron microscope photo shows 0.1 micron diameter asbestos fibers collected from the air of a school. To give you a better idea of just how small these are, let's look at human blood cells at the same magnification. Those big round things you see are individual blood cells. Side by side at the same magnification, you can see that blood cells in 0.1 micron diameter asbestos fibers have a size comparison something like a sewing needle and a water balloon. But what does all this mean? Well, let's take a closer look at a human cell. The cover of the September issue of Science 84 magazine has a particularly good illustration of a single cell. The average size of a cell in the human body is somewhere around 10 to 12 microns in diameter. And when you put a 0.1 micron diameter asbestos fiber next to a 10 to 12 micron diameter human cell, this is what it looks like. But How can something this small hurt us? Well, one way asbestos hurts us is by causing cancers in our bodies. In fact, asbestos is now recognized as one of the most carcinogenic substances on our planet. After being inhaled, asbestos fibers can ultimately cause cancer in a variety of organs, including the lungs, the stomach, the colon, the heart, the chest cavity, the kidneys, the larynx, and other sites and organs throughout the body. Well, let's think about this a minute. Okay, I breathe in 2 million asbestos fibers into my lungs. But my lungs are like bags, and what I breathe inside them is trapped there. How can an asbestos fiber, which is trapped inside my lung, cause a cancer down here, or over here, or up here? How can an asbestos fiber, which is inside the lung, cause a cancer on the outside surface of the lung or in a distant organ? Are asbestos fibers able to move outside the lung somehow? Can they travel to distant organs? Well, yes, they can, and they do. Autopsy studies done on victims of asbestos disease have found that those cadavers had asbestos fibers lodged in tissue throughout their entire body. There were even asbestos fibers as far away as their brain. Medical research over the last few decades has established that asbestos fibers have the peculiar capacity to migrate or move throughout the body. What happens is this: a 0.1 micron diameter asbestos fiber is as sharp as it is strong. And when it gets inside the lung, it has a natural tendency to insert itself between the cells of the lung wall. And then, propelled by the natural motions of living tissue, it begins to move and migrate through those cells. In this manner, asbestos fibers actually move through the wall of the lung, and they can also move through the wall of veins. And when they enter the bloodstream, they ride this vascular highway to all parts of the body. And in the same manner, they can migrate into the vessels of the lymph system, which is another highway throughout the body. And research has established that in females, asbestos fibers can move from the living tissue of a mother into the fetus in her womb. This is called transplacental migration of asbestos because the fibers move through the placenta. The resulting infant is born with asbestos fibers already lodged in its organs. There, they can continue to migrate through that organ. To better appreciate the full biological dangers of asbestos, let's take a closer look at what it looks like when a 0.1 micron diameter asbestos fiber and a single human cell encounter each other. Even without knowing a great deal about cellular mechanics or microbiology, it's pretty easy to imagine the kind of damage this might do. This textbook, Health Hazards of Asbestos Exposure, has a photo of asbestos fibers inside an animal cell, and this gives you a good indication of the real-life size comparison between asbestos fibers and living cells. The dark blob you see at the bottom of your screen is the nucleus of the cell, and the spear-like structures you see above it are 0.1 micron diameter asbestos fibers. The inside of a human cell is a very complicated place which has systems of internal organs much like those of our larger bodies. It's a place of critically balanced electrical, chemical, and mechanical processes which maintain our normal life functions. We know that when an asbestos fiber and a human cell interact like this, something disruptive happens inside the cell. That something could be a chemical, electrical, or mechanical disturbance, or a combination of the three. In any case, medical researchers know it damages the cell in a manner which makes the cell or the cell's offspring ultimately susceptible to the uncontrolled growth we call cancer. And that's why medical and health authorities have become so concerned about asbestos in buildings. Asbestos construction materials inside buildings can routinely release enormous numbers of these fibers into the air to be breathed by building users. And what makes this all the worse is that this kind of contamination is as invisible to the human eye as it can be harmful to the human body. In March of 1985, an American Medical Association journal indicated that airborne asbestos contamination is so prevalent in hospitals, homes, and daycare centers that 50% of the newborn infants examined already had asbestos fibers in their lungs, and that the number of asbestos fibers found in the lungs of these infants is comparable with that found in some adult mesothelioma cancer patients. Dr. Roy Anderson, former director of the Franklin Institute's Advisory Center for Toxic Substances and current director of Comprehensive Chemical Consulting in Newtown Square, Pennsylvania.
That unless steps are taken today, that we will see significant increases in cancer deaths from asbestos by the 1990s, and in the future, and beyond the 1990s, until the asbestos problems have become corrected. So this is a significant public health disaster that is brewing. The problem is that when you have been exposed to asbestos, nothing happens for perhaps 20 or 30 or 40 years. And during that quiet time, the material is working its way through your body and starting to begin the cancer-causing process, and you have absolutely no way of knowing whether or not you're going to come down to it. I sometimes wish that when we were humans were exposed to asbestos, that somehow or another we would turn green or blue immediately so that we know we'd have the asbestos exposure and then possibly could do something about it. But at this point, there is nothing that can be done. When the asbestos-related diseases develop, the only thing that physicians can do is to reduce the pain. That's it. They can't cure it.
So if you own, live in, work in, or otherwise utilize a building, public or private, how do you know if it has an asbestos problem? This is a question that raises very serious legal, health, and technical issues, and the first thing you should do is find somebody who knows what they're talking about to advise you. This could be an architect, it could be a toxic substance consultant, or it could be a professional asbestos survey firm. Whoever it is that you get, you should be absolutely sure that they have in-depth knowledge and experience with the asbestos in buildings problem. In Ardmore, on Philadelphia's main line, architect Paul Getz has specialized in asbestos in building work for a decade. He has been the asbestos control authority retained by such retail conglomerates as Allied Stores of New York and other institutions such as the U.S. Postal Service headquarters in Washington and CBS TV.
It's important for a building owner to have a accurate evaluation of the friability. It's important to have the proper testing done, and it's important, if an asbestos situation does exist, that the custodial, the maintenance personnel are instructed on the proper procedures relative to working with or working around asbestos content materials. And what we have here's a office building in upstate North New Jersey. We walked in, found the typical acoustical panel ceilings, and doing the research we got up and into the ceilings. This is what we found. The structural steel frame was sprayed with very friable material. Friability is the key word, whether it's presenting a potential hazard. In this instance here, this ceiling, this plenum was used as a return air plenum, so the air was being circulated, returned from the room area to this plenum, taken up to the air handling units on the roof, and recirculated back into this office for the occupants of this office to inhale the asbestos fibers.
In order to make an accurate judgment about the asbestos hazards which might be present in an office building like the one you just saw, or an industrial complex such as this one, you need to know two very important things. One, are there asbestos-containing materials present anywhere in the structure? And two, are those asbestos-containing materials friable? That is, are they releasing fibers into the air? Well, the U.S. Environmental Protection Agency for years has been telling the public that one of the best ways to learn these things is to use blueprints, to dig out the construction blueprints which were used to build your building and see if the architect back then specified asbestos-containing materials. However, authorities in the field with long years of experience in studying buildings for asbestos warn that using construction blueprints like that is not accurate. Architect Paul Getz.
Regarding a building owner doing asbestos research on their own and making a study of the construction drawings of their building, they could be very much misled if they just believe what's on the construction drawing and not going out into the construction itself and finding out if there is asbestos content materials in the building. A lot of cases that we've come up against where an owner has handed over to us their construction drawings, we find that the construction drawings do not agree with the conditions that we found when we have gotten up and into the constructed buildings.
When surveying a building for asbestos hazards, two very different kinds of samples are collected. The first kind is called a bulk sample. The second kind is called an air sample. And it's the combination of information that you get from both the bulk samples and the air samples which determine whether or not you have a hazard in your building.
Bulk samples take their name from the fact that you actually collect a bulk, a piece, a chunk of the suspected material. Here, I've collected a piece of suspect insulation from a duct above this ceiling, and I place the insulation in a plastic jar, or I might place it in a plastic bag which could seal tight. You'll be collecting tufts of insulation like this, or slices of pipe covering, or plugs of coated ceiling surfaces, or corners of acoustical tile, or perhaps pieces of linoleum tile. Anything that you suspect might have asbestos in it, you actually take a bulk or a piece of that item and put it in a sealed container. However, this isn't something you should do casually or without a good deal of thought. Dr. Roy Anderson explains.
For example, a building owner could get into an awful lot of trouble, and a lot of school districts have gotten into a lot of trouble by trying to take samples. Taking bulk samples is not as easy as it looks, and unless you take care and take pains to protect yourself or the person doing the sampling, you can be exposed to significant levels of asbestos fibers while you're doing the sampling. The other problem, of course, is that you need somebody who has at least had some experience in this field and who is reputable and is not a opinion. There are, unfortunately, too many instances of building owners being told that there is asbestos in their building when in fact there isn't. And the material that was suspected of being asbestos turned out to be cellulose. And they have spent thousands, hundreds of thousands, or millions of dollars in a completely needless removal of relatively harmless materials.
Keeping good records as you collect samples throughout a building is crucial. You should be as comprehensive as possible, and remember, you are creating what could become an important, legally relevant document. You should use hand-drawn maps or blueprints and mark the exact spot from which a sample was taken. And you should at least record the date, the time, the location, and a description of the type of materials collected. Then, the bulk samples are ready to be delivered to a laboratory for analysis. At the lab, a technician will take a portion of the material, magnify it, and analyze it for asbestos content. But, like so much else about asbestos, this process is somewhat controversial. For years, the U.S. Environmental Protection Agency has advised building owners to use polarized light microscopy, or PLM, to analyze bulk samples. And for an equal number of years, asbestos safety authorities across America have been reporting that the PLM method is not very accurate. Many, in fact, now ignore EPA guidelines and have switched to using either SEM, scanning electron microscopes, or TEM, transmission electron microscopes, for this purpose. Structure probe laboratories are a series of research facilities located throughout the eastern portion of Canada and the United States. And during the last 10 years, they've been heavily involved in studying asbestos construction materials used in schools and other buildings. Dr. Charles Garber, president of Structure Probe, is nationally renowned as an authority in the field of asbestos analysis.
Once you have done your sample collection, now the question is, how are you going to do your analysis? The most commonly used method is called polarizing light microscopy, or sometimes dispersion staining, which is a specialized form of polarized light microscopy, oftentimes abbreviated as PLM. This method is by far the lowest cost. It is the fastest way to get some type of analytical data. However, because of the complexities of the structure of many insulation materials, the method is not good enough to really be used as a sole method to determine whether there is or is not asbestos present. That in our laboratory, we use a combination of PLM and X-ray diffraction. That if indeed we do get results that between the two methods are consistent, then we believe that we do have the correct answer and report it as such. But there are times when the two approaches do not agree, and consequently, in order to determine what really is present in that kind of a case, further analyses are then necessary.
After its analysis, the laboratory provides a report which indicates how much, if any, asbestos was found in samples like this one. The report might indicate, for instance, that the technician found 18% amosite in a pipe covering or 23% chrysotile asbestos in a ceiling spray. Remember, bulk tests only confirm that asbestos materials are present in the building. They don't necessarily indicate whether those materials are posing an immediate airborne contamination danger to building occupants. To make that determination, a second kind of test, an air test, is performed. The Micromax pump is especially designed for asbestos air testing, and it's used for drawing a specific amount of air through a cassette which contains a special filter material. The filter in this cassette traps and holds particles which are suspended in the air and drawn through it. In this way, we're able to collect a sample of the particulate floating in the air and analyze it to see how much asbestos it contains. As you can see, the cassette has plugs at either end, a red one and a blue one, and it's made in sections. You can twist off the front, there's one section. You can twist off the second one, there's the second section. And now what you're looking at inside, this white material, is actually the filter itself, and it's so delicate. It's called a membrane, and I think you can see why. This is an extremely delicate kind of filtration material. And the holes in this thing are so small they're invisible to the human eye, and as a matter of fact, the way they're made is by bombarding this material with neutrons, atomic particles, which drill holes in it. If we take this out and we put it under a scanning electron microscope and we blow this filter up 6,000 times, this is what the surface actually looks like. When we pump air through such a filter, particulates floating in the air are trapped on its surface, like this. This is the actual filter from a recent building air test. The things which resemble boulders are the kinds of airborne microscopic debris normally found in the air. The long, thin structures you see are asbestos fibers which were floating in that building's air and being breathed by that building's occupants. To actually begin the sample, first we turn on the pump. Then we use the controller knob to set the exact rate of flow, which in this case will be 15 liters per minute. Then we take our air test logbook and we log in some crucial information. One, we put the name of the person who took the test, the date this test is being taken, the location at which the test is being taken, the rate of flow we are using on the pump, and the time that we turned the pump on. In this case, 12 noon. And now I have about 3 hours to kill while this pump runs and collects 2,800 liters for our air test. Hello again. It's now 11 minutes after 3 o'clock in the afternoon, and our pump has been running for 187 minutes. So 2,800 liters of air have now passed through the filter in this cassette, and our sample collection is finished. We can turn off the pump. Immediately after turning off the pump, we have to record the exact time that we turned the pump off in our logbook. Then I prepare a label which shows the date, the total number of liters collected, and other identifying information about this actual sample. This label is then placed on the exterior of the cassette, and from now on, I and the lab will always know what this cassette is. So, this cassette now contains a representative sample of the particulate matter in the air of this office building. And the next step of the process is to take this cassette to a laboratory. There, a technician will cut out a small portion of the membrane filter, put it under a microscope, and analyze it. Now, this process isn't as simple as it sounds because there's something of a controversy about what kind of microscope should be used to analyze air samples like this one. There are 3 different kinds of microscopes involved in this issue: the traditional optical or glass-lensed microscope, the scanning electron microscope, and the transmission electron microscope. Up until the 1950s, the only magnifying tool available to analyze anything was the traditional optical microscope. Instruments such as this one can be found in thousands of high school, college, and commercial laboratories across the country. Although optical microscopes come in a variety of sizes, shapes, and colors, their basic features really haven't changed much in the 300 years since the instrument was invented. All optical microscopes are essentially a hollow tube with glass lenses fitted at either end. As a matter of fact, the reason they are called optical microscopes is because they employ optical glass. They are also referred to as light microscopes, polarized light microscopes, PLM, phase contrast microscopes, and similar names. In these optical or light microscopes, visible light rays strike a specimen and are reflected into the tube. There, the light rays are bent by the glass lenses in a manner which results in a magnified image at the top. For reasons that have to do with complicated principles of wave physics and the limitations of glass, optical microscopes are very limited in what they can see. For instance, they can't see anything much thinner than about 0.5 micron in diameter. When you put something that thin under an optical microscope, the rays of light and the glass lenses can't resolve it into a visible image, and so the human eye can't see it. This was a serious problem for many, many decades to researchers, and by the 1940s, scientists in both the United States and Europe had developed a completely new kind of magnifying device. It didn't use rays of light, and it didn't use optical glass lenses. Instead, this new instrument used electrons. Electrons are particles of atoms which are released when atoms are excited in certain ways. For instance, when you run electrical current through a tungsten filament, large quantities of electrons fly off in all directions. However, if you put this filament inside a vacuum chamber and use magnets, you can draw the escaping electrons into tightly focused beams. In fact, magnetic fields can be used to manipulate electron beams the same way that glass lenses can be used to manipulate rays of light. And if such a beam is passed or transmitted through a thin specimen, some electrons will be held back by the thicker parts of the specimen. When the resulting electron pattern is translated into visual images, a one-dimensional picture of the specimen can be seen. Because the beam is transmitted through the specimen, this machine was called a transmission electron microscope, or TEM. In the 1950s, scientists developed a second kind of electron microscope. This one also used electron beams, but it created images in a different manner. Electron beams were generated, focused by magnetic fields, and then scanned back and forth across the specimen. This technique employed the same electronic scanning mechanisms utilized in standard television tubes. The electron beams did not pass through the specimen, but rather reflected off its surface. These reflected signals were gathered and translated into three-dimensional pictures on a television tube. This machine was called the Scanning Electron Microscope, or SEM. The levels of magnification possible with such electron microscopes were enormously greater than those of optical microscopes and provided stunning new views of our world. Both the scientific and the popular press hailed the electron microscope as one of the major technological milestones of the 20th century, and they continue to report how the instruments are revolutionizing every aspect of science and human knowledge. One of the areas of knowledge which was vastly expanded by the use of electron microscopy involved asbestos. As early as 1950, scientific journals reported that researchers were using prototype electron microscopes to study human lung tissue samples, and what they were finding were asbestos fibers which were so tiny they had been invisible to previous researchers who had only had optical microscopes. In 1958, for instance, the Johns Manville Company publicly indicated that its electron microscope researchers had discovered previously invisible asbestos fibers. And Johns Manville went on to report that such fibers were actually as strong as some types of steel wire. To get an idea of the sort of new information that was emerging back then, let's look at some photographs taken on a scanning electron microscope. This is a strip of air sample filter like the one you saw me collect earlier, magnified in an electron microscope 48 times. The large white object you see is a pinpoint we've put on the filter surface for size comparison. The white material covering the rest of the surface is particulate collected from the air. Now let's increase the magnification to 480 times. This is a very important number because 480 magnification is the maximum available on the standard optical microscopes which are used for asbestos analysis. What you're looking at is the smallest kind of asbestos fibers visible to optical microscopes. And up until the 50s and 60s, this is as far as researchers could see. However, when electron microscopes became available, this is the new world of vision which was opened on asbestos. This is a magnification of the same filter surface 3,200 times, and as you see, this filter surface is blanketed with thin asbestos fibers which were totally invisible at lower magnifications. And earlier in this program, we showed you how fibers of this size compare with a human cell. So, if you're in any way involved with testing buildings for asbestos hazards, it's crucial to keep in mind that different microscopes have different limitations. Let's look at what world authorities have published on this subject in recent years. In its landmark 1973 publication on asbestos and cancer, the World Health Organization's International Agency for Research on Cancer reported, in man, a proportion of the fibers that may be carcinogenic are not seen or counted by optical microscopes. In 1976, the National Institute of Occupational Safety and Health, in conjunction with the Center for Disease Control, reported that many airborne asbestos fibers are generally below the resolution limits of the optical microscope. These fibers may only be detected by using electron microscopic methods. In 1978, the National Cancer Institute reported that when analyzing the air for asbestos contamination, the majority of fibers will be below the resolution of the optical microscope. In 1979, the Journal of the American Industrial Hygiene Association reported that electron microscope analyses of air samples revealed many asbestos fibers which were too fine to be detected by an optical microscope. Electron rather than optical microscopes should be used for air sample analysis. In 1980, the World Health Organization's International Agency for Research on Cancer held another International Asbestos Seminar, which it said, confirmed again that a considerable proportion of fibers escape detection by optical microscopes. In 1983, when the U.S. Environmental Protection Agency issued new guidelines on controlling asbestos in buildings, it reported that optical microscope methods have major shortcomings and could be highly misleading. Shortly after that, a top EPA official testified at Washington asbestos hearings that The number of small fibers in the air is much greater than those visible to optical microscopy and may present a significant risk by being present in much larger numbers. Despite this kind of documentation stretching back for decades, in 1985, as we are making this film, federal agencies such as the EPA and OSHA continue to allow school districts, industrial facilities, and public and private building owners to use optical microscopes to determine if their air is free of asbestos and safe to breathe. To fully appreciate the implications of this governmental policy on the breathing public, let's look at some actual comparison tests between optical and electron microscopes on air samples from New Jersey buildings. This is the surface of a nuclear filter from an air sample collected by the Dual Corporation in a New Jersey school building. This is a scanning electron microscope photo at 480 magnification, so it's comparable to the best that an optical microscope could see while analyzing this filter surface. As you can see, there are clumps of normal airborne debris, but no apparent quantities of asbestos fibers visible at this magnification. And under the prevailing federal policies, this school building would be declared safe and reoccupied by thousands of children, maintenance workers, and teachers. However, you see we've put a little white box in the center of the filter. Well, if we magnify the area within that box 6,600 times by scanning electron microscope, this is what we see. The air in the building is glutted with airborne fibers, totally invisible to the optical microscope. This building is not safe, it is very hazardous, and it requires extensive decontamination, but you wouldn't have known that unless you had used an electron microscope for analysis. In September 1983, the respected national educational journal American School and University warned the nation's schools that the optical testing methods allowed by the federal government were highly questionable, and that increasing numbers of private asbestos safety authorities had quietly stopped following the vague and confusing government guidelines in this area. Dr. Roy Anderson, Director, Comprehensive Chemical Consulting.
The optical microscope cannot distinguish between asbestos and other fibers, and it cannot, more importantly, the optical microscope does not have the proper resolution to see the very fine asbestos fibers that could be present in the air and that are typical of deteriorating asbestos containing materials in buildings.
Anthony Natal, president of Dual, one of America's oldest asbestos control firms.
Well, from what we've gathered in the government studies, their studies themselves show that the optical instrument is inadequate to do the job on asbestos. The information that we've gathered through our testing also confirms that. We've brought this information to the government, EPA, OSHA, NIOSH, the state government, and all agree that the optical instrument is not the instrument to use for counting fibers, asbestos fibers, and to make sure that the environment is clean of these fibers. The problem is they still recommend the use of the optical instrument. Why, I don't know.
Paul Getz, architect.
A researcher or an owner can't hide their head legally under the carpet by abiding only by the EPA, OSHA regulations or guidelines. It's important to know that legally, if a technique of the state-of-the-art is known, you have the legal responsibility to use that technique, that state-of-the-art, and not just abide by the current guidelines and regulations of your governmental authorities.
At the Hitachi Nissi-Sanyo Laboratories in Rockville, Maryland, we can get a close look at one of the instruments which is being used for state-of-the-art asbestos testing, a Hitachi 570 scanning electron microscope. Scanning electron microscopes like this one are the product of space-age high technology, and they represent a creative merger of computer controls and electron optics. Not only can this instrument routinely detect the thinner kinds of asbestos fibers, but an attached computerized analysis machine called an EDX can identify most asbestos fibers by their elemental makeup. And so, this equipment not only tells you if asbestos is present, but the type of asbestos that's present in the air in your building. The best way to understand how this system works is to actually see it in operation. Here at the Hitachi lab, Mr. Sunio Akiba will show us how an asbestos air sample is processed. First, in a laboratory clean room, the cassette is opened and a strip of its filter is cut out. Then, the strip is mounted on a metal stub. This stub is then electronically coated with a thin layer of gold in another laboratory device known as a sputterer. The gold coating makes the specimen much more reactive to the electron beam of the microscope. This coating process takes only a few minutes. Now, the coated stub is ready to be placed in the vacuum chamber of the scanning electron microscope. For our purpose today, we're only going to load one sample stub. However, in an actual laboratory production situation where many samples must be processed each day, a dozen or more sample stubs would be loaded into the chamber on a larger multiple stub carrier. Then a vacuum is restored to the chamber and the scanning electron beam is turned on and adjusted with the various computerized controls. The magnified surface of the filter comes into clear view as a three-dimensional television picture. Fibrous shapes are easy to see and can be magnified tens of thousands of times with the push of a button. An additional feature allows the operator to target a fiber for X-ray microanalysis. The results of this appear on a nearby terminal and show what elements are contained in the targeted specimen. In this way, fibers can be identified as asbestos. State-of-the-art scanning electron microscopes like this one can see very small particles of asbestos. This is the surface of a millipore air filter sample at 1000 magnification. A touch of a button brings the filter surface to 5000 magnification and previously invisible fibrous forms come into view. Another touch of a button increases the magnification to 15,000 and an asbestos fiber of 0.1 micron in diameter can be seen in detail. And with a final touch of a button, magnification is increased to 50,000 times and we can see the thinner strands which make up that fiber. We are now looking at objects which are smaller in diameter than 0.05 micron. And this is the beginning of the size range for fibrils, the final, indivisible, elemental particles of asbestos. And while scanning electron microscopes are powerful, the next machine we're going to look at is even more powerful. At Structure Probe Incorporated's laboratories in Westchester, Pennsylvania, we're going to see how a transmission electron microscope is used to analyze asbestos samples. Transmission electron microscopes require more tedious sample preparation and are more complex and more expensive than scanning electron microscopes. They are also so powerful that if you wanted to, you could actually study the molecular structure of asbestos or any other material. This particular model is a 100CX made by JEOL Limited, whose American headquarters are in Peabody, Massachusetts. Let's see how it works. The filter cassettes, such as the one we used earlier to collect our air sample, are logged in and brought to the special preparation room. A lab technician opens the cassette, takes out the nucleopore filter membrane, cuts it in half, and places one half on a glass slide. This slide is mounted on a paper wafer which holds it in place in this special Petri dish. The glass dish protects the filter sample as it is taken to another section of the laboratory to be coated with carbon. The slide is placed in the coating machine. Physically, a thin layer of carbon is applied to the filter surface. This carbon coating is similar to the gold coating we saw a moment ago in the scanning electron lab, and it serves a similar purpose by making the sample more conductive during exposure to the electron beam of the microscope. The coated filter section is brought back to the preparation room and chloroform is added to another Petri dish nearby which holds the special metal grids on which sections of this filter will be mounted. The filter is cut into small squares and the squares are then picked up and placed on one of the metal grids in the chloroform treated dish. This dish is then put aside for 24 hours, which is how long it takes for the chloroform to dissolve the filter material. Because in this type of microscope the electron beam must pass through the specimen, the filter must be dissolved away. After the 24-hour waiting period, the Petri dish is brought back to the microscope room and the prepared filter sections are loaded into the microscope's specimen carrier. The specimen carrier is inserted into the column of the instrument and seats itself automatically. The microscope operator can then begin the procedure of analysis. The various controls bring the specimen into focus and then adjust the magnification to the desired level. A projected image becomes visible in the viewing area of the microscope and allows the operator to study the contents of the specimen grid by grid. This specimen, magnified about 15,000 times, is what a typical asbestos air sample looks like in a transmission electron microscope. The fibers which appear in such samples can be then subjected to what is known as electron diffraction analysis. The beam of electrons can be made to pass through a single fiber, and under the right conditions, the fiber produces a specific pattern. Because all crystalline materials have unique electron diffraction patterns, the operator is able to identify the fiber by this signature or fingerprint. This particular pattern shows the fiber to be chrysotile asbestos. After your air filter cassette has been analyzed at the lab prepares a report to tell you what they found. And that report may use one of two different kinds of units of measurement to describe the asbestos conditions documented by the technician. The first form of measurement involves an average number of fibers found in a given airspace, mostly smaller than my thumb. A second form of measurement tells you the total weight of fibers found. This measurement is given in nanograms, which represent 1/1,000,000th of a gram in weight. This is a very exotic form of measurement which has no real meaning to most people. From the point of view of a layman trying to make intelligent decisions about health conditions in a building, the first form of measurement, an average number of fibers per cubic centimeter, makes more sense. You'll frequently see this abbreviated as F/cc. And this same measurement is also referred to as a fiber count. The lab technician uses the information he collects under the microscope to compute the average numbers of fibers floating in the airspace that was sampled. So, the lab might report to you that your sample indicated a fiber count of maybe 1.5 fibers per cubic centimeter. This means that an average of 1.5 asbestos fibers are floating in every cubic centimeter in the airspace where you're breathing. But what does this really mean? Well, let's step over to our other set for a minute and look at a cubic meter in the human lungs. This is a cubic meter. It's 1 meter wide by 1 meter high by 1 meter deep. And the air inside this box is about the same amount of air that we breathe every hour. That is, the average human being breathes in and out about 1 cubic meter of air every hour. And that makes the cubic meter a very convenient unit of measurement for computing how many asbestos fibers we're breathing in any given airspace. For instance, let's say that our lab has just told us that our air sample shows 1.5 asbestos fibers per cubic centimeter. Well, we have to convert that into some meaningful form for laymen. And the way we do that is to take centimeters and convert them into cubic meters. Now, 1 meter happens to contain 100 centimeters. So this box is 100 centimeters wide, 100 centimeters high, and 100 centimeters deep. And with some simple arithmetic, if we take 100 times 100 times 100, we'll discover that there are 1,000,000 cubic centimeters in a cubic meter. That sounds like a lot, but remember, a cubic centimeter is hardly the size of the tip of my thumb. And the easy formula for converting fibers per cubic centimeter to fibers per cubic meter is to take the fibers per cubic centimeter and multiply that times 1,000,000. In this case, let's take 1.5 asbestos fibers per cubic centimeter and multiply that times 1,000,000, and that tells us that within 1 cubic meter of air in that airspace there are about 1,500,000 asbestos fibers. That means that in that airspace all the people there are breathing in about 1.5 million totally invisible carcinogenic asbestos fibers every hour. So as you can see, a reading of 1.5 fibers per cubic centimeter is certainly not a safe level. And that brings us to the question of what is a safe fiber count, and unfortunately there's no easy answer to that question. Medical authorities studying patterns of asbestos cancer now warn that there is no safe level of airborne asbestos contamination. The medical evidence shows that any level of asbestos contamination is likely to increase the number of cancers in exposed populations. But we live in a land which is virtually awash in airborne asbestos contamination. Even the air on our city sidewalks has significant levels of asbestos in it, so the question really is, what minimum level of airborne asbestos is reasonably acceptable in our buildings? In March of 1984, asbestos safety experts from North America and Europe assembled at the National Bureau of Standards in Gaithersburg, Maryland, to discuss this question. No conclusions from this meeting have ever been published, but the general consensus expressed by participants was that an airborne fiber level of 0.01 fibers per cubic centimeter, as measured by an electron microscope at 10,000 magnification, represented a level which could be routinely achieved with currently available decontamination technology. Many people are often tempted or erroneously advised to use OSHA's asbestos air standard for making judgments about how safe a building's air is. And as this film is being made in 1985, OSHA's asbestos air standard continues to be 2 fibers per cubic centimeter of air. This means that OSHA allows employers to expose their workers to 2 asbestos fibers per cubic centimeter of air every day, day after day. This is a widely discredited standard. For instance, in 1979, NIOSH assembled a committee of experts to analyze the OSHA air standard against the currently available medical data about asbestos cancer. In a memo explaining the project's findings, the director of NIOSH explained, "The committee concluded that the current 2 fibers per cubic centimeter standard is grossly inadequate to protect American workers from asbestos-related disease." In 1982, the New York Times reported that OSHA's own technical experts had conducted an internal study to assess the death risks associated with the agency's current standard. The Times said that OSHA researchers had concluded that at the current OSHA standard of 2 asbestos fibers per cubic centimeter of air, 8 to 260 workers per 1,000 would develop lung disease. In 1983, an OSHA asbestos risk assessment report was published in the Federal Register. It said, 2 fibers per cubic centimeter is equivalent to 2 million fibers per cubic meter of air. Humans breathe in about a cubic meter of air every hour. The current OSHA standard for asbestos allows workers to inhale 2 million asbestos fibers per hour. And keep in mind that OSHA exclusively uses optical microscopes to analyze the air. When OSHA finds 2 fibers, that only means that OSHA has found 2 fibers visible to the optical microscope. For instance, this millipore air filter sample collected in an office building would register as clean under OSHA standards. But when a scanning electron microscope is used to magnify the filter surface 4,200 times, vast numbers of asbestos fibers which were invisible to the optical microscope are revealed. These are the kinds of conditions which routinely exist in air spaces which meet OSHA standards. Clearly, OSHA standards represent a very dangerous level of exposure. OSHA standards do not now, nor have they ever been based upon serious health or medical considerations. OSHA standards represent political accommodations between multi-billion-dollar industrial lobbies and the political establishment in Washington. No reasonable person in good conscience could ever apply OSHA standards to schools, offices, or other public buildings. To do so is to place the occupants of those buildings at grave risk. And that's going to wrap up this segment of Asbestos Safety Workshop. We'd like to thank you for being with us today.